So this is one of our student workers, Audrey Maddox, and she is currently observing 1 meter squared of blueberry blossoms to see if there are any bees visiting. We do this at distances, at set distances from this wildflower planting. So this is the closest distance. And I don't know if you can see, there are 2 more flags that way, also at the close distances. And then we'll also do the same thing as we're doing here at each flag at different distances. So we have a 0, we have about a 10 and a 20 and a 60 meter distance. And so we have a total of, the idea is to have a total of 24 observations for each site during blueberry bloom. Because any one visit, you might get nothing. There might be nothing there in that minute where you're observing that meter squared, or you might get 15 bees. That'd be a very high number. So it really depends, and a lot of it depends on the weather. So when we do this, we also record the weather so we can see if that's how much of the variation the weather is accounting for, temperature, cloudiness, etc. We have the wildflower and clover, and over here we're testing just a natural regeneration. But the reason we're doing it is to see which one of these is more attractive and has the most potential to enhance the bee community. So here we have 3 species of clovers that are all planted together, and then the natural regeneration was tilled, and then we're just allowing whatever was in the seed bank to come back. So we're also doing diversity samples. So we're trying to determine what the bee community is like in different fields. We have fields with the wildflower plantings, and then we have control fields that don't have the plantings. And so twice a season we go out with these, and we collect bees. 2 people collect bees for a total of a half an hour. And we usually get anywhere from 20 to 30 specimens. And then we can take those bees into the lab and under a microscope identify them to genus or species. And then we can use a diversity metric in order to compare how diverse the community of bees are in the control fields versus the treatment fields. And the idea is that hopefully in these fields that have more abundant forage because of these wildflower plantings, that there would be a more diverse community of bees. And maybe also then we'd see some benefit in terms of pollination in the blueberry field. So we collect the samples once right around bloom, towards the end of bloom, and then we collect samples again in August to get a better feel of the community over the entire season. So briefly, the bumblebee life cycle, let's start in the fall actually. And so a colony is sending out females, they're called gynes in the fall, these future queens, and also males that will mate with the queens. So then a mated queen, a gyne, in the late fall will actually leave the colony, burrow underground, and overwinter, go into a suspended state. And in the spring, these same queens emerge, and these are the big bumblebees that you see early in the season that are out poking around in stone walls and in brush piles. And they're not looking for flowers in that case necessarily. They're looking for a place to start a colony. They're looking for a nice place to nest. And once they do start a colony, then they start to forage for pollen and nectar and use the pollen and nectar to feed their first generation of workers. So they rear their larvae, and then the first thing you see after you'll see the large queen bumblebees buzzing around are the very first tiny workers in the spring. And they can be so small that you might even confuse them with other bees because they're much smaller than most people would consider bumblebees to be. So when we do our first round of sampling, we could collect queens, but because those queens are going to start a colony, it would really negatively affect the population of bees on the farm, and we don't want to influence the data in that way, nor do we want to affect the bee community in that way because the farm is relying on these bees. But we collect bumblebees and we take their pollen loads, and then we analyze the pollen loads under a microscope, and we can determine the proportion of different flowers that have gone into that pollen. And so we do that for 15 bees at each field between control fields that don't have a wildflower planting and fields that do have a wildflower planting, and we can compare what they're using to feed their larvae in terms of pollen. And so the idea is that in fields with the wildflower planting, that the bees would be using more of these planted flowers, and we could get a direct measure of really the utility to the bees of these planted flowers. Do you have any bees in there?
A honeybee and a bombus.
Great. We've got a bombus here, probably a bombus in patience, and a honeybee.
Hi there, my name is Audrey Maddox, and I am a fourth-year undergraduate at the University of Maine. This summer I will be researching squash, particularly winter squash in Maine, and I will be examining the bee community visiting squash to really see what native pollinators are there. I'm particularly interested to see whether the squash bee, Peponapsis prunosa, has spread this far north, like what its range is, because we haven't documented officially whether it is present here in Maine. So that's what I'll be doing this summer.
Well, currently the only bumblebees that we have are commercially produced non-native species from Michigan. And we hope that in our experiment that we'll have a native species to use because it'd be more applicable in a Maine setting. So these queens are of the orange-belted bumblebee variety, and they're very common in Maine.
Well, why don't you tell our audience here what you're doing with the experiment that you're setting up with the commercial bumblebees.
Well, with the commercial bumblebees, I have quite a few of them right now. I'm dosing them with different doses of imidacloprid, which is a neonicotinoid, which has been in the news a lot lately, and looking to see if there's any effects on the colony's development and growth. So I'm looking at weight. And then I'll be doing that for another 2 weeks, and they'll be set out into Lowbush blueberry fields throughout the summer, and I can check on them throughout the summer. And then at the end of the season, I can look and see their production of reproductives, as well as if they're more prone to get parasites or pathogens. This chamber is kept at a constant temperature of 20 degrees centigrade, so 68 degrees Fahrenheit, with a 55% humidity and with a light day cycle to keep the bees on a realistic cycle. Here are my commercial colonies of bumblebees. This species is called Bombus impatiens. And then down here I have a collection of my wild-caught queens that I caught around fields in Maine that I'm trying to rear and get to lay eggs and start their own colonies. That's the typical Bombus ternarius, or the orange-belted bumblebee. And then I have a couple other species in here as well. So first what I do is I weigh the entire colony. So this includes all the bees and their brood. And I also gave them a little bit of pollen, these pollen balls that I made out of ground up pollen and feed to provide extra food for them. So after that is weighed, I also monitor their feeding levels. So all these bags are weighed every day. My findings so far, this is one of my control colonies, so there's no imidacloprid in this bag right now. But basically, the control colonies are gaining in weight, and this increase in weight gets smaller and smaller as the dosage goes up, and actually there's a loss of weight in the highest dose in my colonies. So that's an interesting finding so far, because this experiment's only been going on for a week. And with the bags, the bag weights have been going down more steadily in the lower doses, meaning that they're feeding, whereas in the higher doses, they're not feeding as much, and that might be due to an aversion of the imidacloprid, which has been found in some of the literature.